We are here with Lars, also from the DSMZ in our MICROBE consortium. Thank you that you take your time to give us a short interview. And yeah, we start right away. Maybe you give us a short introduction, who you are, where you're from, your background, a little bit about your scientific career up to now, when you came to DSMZ and the MICROBE project.
Yeah, thanks. My name is Lars Möller. So I'm here at the DSMZ, the German Collection for Microorganisms and Cell Culture, and I'm currently working in the MICROBE project. But my scientific career, I can say, started actually in high school. At this time it was still mandatory to do an internship in a company or institute of your choice, and at that time I chose to work in the Museum of Natural History in Berlin, the Leibniz Institute for Evolution and Biodiversity Science. And during that time it really encouraged me to pursue studying biology, which I did then in Rostock. And during the bachelor you get a broader idea what biology really means, and at that time I really took a liking in microbiology and biochemistry. And therefore I continued by studying this topic in my master thesis. And after that I did my PhD in marine microbiology at the IOW, the Leibniz Institute for Baltic Sea Research in Warnemünde. And after that I continued with my first postdoc in Oldenburg at the university there, which brings me now here after the 3 years to the DSMZ and the MICROBE project. So this is basically how my whole process of science work started and continued.
You already mentioned the MICROBE project, so that you explain short what are you doing in a MICROBE project, or how did you came into the project team here, and what are your main goals for yourself also scientifically from the MICROBE project?
Yeah, so my part in the MICROBE project is the preservation of complex microbial communities. So we test different methods to see which one works the best, as well as to isolate keystone species of the soil, so organisms which play crucial roles in biochemistry or in maintaining the soil as itself. So this is also my two main goals, to understand this a little bit better, and also to learn the new techniques which we can do here. So the DSMZ is a very interesting institute because it gives you a lot of opportunities to approach your goals with different methods, and I want to learn as much as possible. This is also why I choose this position, because also the DSMZ is one of the main institutes in Germany for studying microbiology. And when this position was open, it was basically straightforward for me to apply for this position.
You were successful with the application, and that's good that you are in our consortium. Maybe you can further elaborate a little bit why MICROBE, or why you think MICROBE is so important for you or your team or the organization also.
Yeah, so in general the MICROBE project is very interesting in itself, because this whole idea of maintaining microbial communities as they are for a longer time period is something which is relatively new. So far mostly single species were preserved, and this is an interesting topic, as well as the whole keystone concept for microorganisms is relatively new, and this makes this so interesting also for me from topic-wise. Furthermore, here at the DSMZ, where we conserve strains, it would just broaden our possibilities to build up collections, not only for single organisms but for communities in total. And this is so valuable for the project, not only for the topic itself and for everybody, but also for the DSMZ.
What are your expectations, let's assume in 3 or 5 years, or after the project, for yourself in this developments that you described, or your personal development, and maybe also the project and follow-up work?
Yeah, so for myself, I basically hope that I can learn as much as possible, also to collaborate with so many partners, so many institutes together, so that it will basically lead me to the way also to become somewhat of a work group leader myself. So this is hopefully where I end at some point. So leading a small working group and then using this knowledge I get here to basically achieve this goal. And I think I have the opportunities with this project here. Knowledge we gain here can really help the scientific community to work on complex communities, to preserve them, and not only in soil. Other group partners, for example, work on the marine system and also in seeds. And what I really hope that is also one goal here at the MICROBE project is that we are able to build up synthetic communities. So using single microorganisms, put them together, and basically replicate what nature at the moment is capable, and this can be useful for other purposes, for example also like applicational service as well.
I assume also that you're not only working here, not only for MICROBE and DSMZ, you also have other life. Maybe you can give us some insights in your other life.
Yeah, so basically besides science I do a lot of sports. So I do team sports in form of volleyball, so 3 times a week, because basically it relaxes me. Often science is more of a mind focused work, and to basically balance this out I do the sports, so that both sides basically get exhausted at the end of the day. Yeah, and also I do fishing, for example, like in my free time, as well as yes, a few bit of hiking, yeah, and meeting friends.
Yeah, thanks very much. So I think you have a good life. And, so thanks very much for your time. It was a nice interview, and yeah, I wish you all the best for your future. Also that you reach the goals in the project, and yeah, good that you are in the team. Thank you.
Thank you very much.